Okay, in this video tutorial I'm going to talk to you about DNA structure. So let's have a look at what this DNA thing is. Before I get into this, for those of you who watched the two little video clips that come before this, if you're watching this as a playlist, you will have seen a little video that explains, the history kind of leading up to this. So there was a time essentially where they knew that every living cell that had a nucleus, inside that nucleus was a chemical substance called DNA, deoxyribonucleic acid. I'll write that up in a sec, which had 4 different types of bases in it. And it was later came a piece of research by a guy called Chargaff, who basically discovered that of these 4 bases, so these 4 different types of bases, adenine, thymine, cytosine and guanine, there was always these ratios where the number of adenines and thymines, A's and T's, was always the same, and the number of C's and G's, cytosines and guanines, was always the same as well. So that gave a clue to the structure of DNA, and so just keep that in your mind as we go through this, and I'll make reference to that again. So DNA is deoxyribonucleic acid, so we should probably write that up. Let me just move across a little bit. So deoxyribonucleic acid. Now you may, in some textbooks, see it written as, and I have a habit of doing this myself, you may see it as deoxyribose with an SE, which is actually what it was first referred to by the 2 people who actually discovered it. So this has actually just been slightly shortened here. So deoxyribonucleic acid. You may see in some books or in some places it referred to as deoxyribose, so BOSE nucleic acid. Both of those, as far as I'm aware, are acceptable. This is probably the slightly more modern version. So deoxyribonucleic acid. And that is a question that can be asked in tests sometimes. So you may be required to actually know the name. Okay, so let's have a look at what is this deoxyribonucleic acid stuff? Like what is it? What is it? What is it made out of? What does it look like? Most of you are probably actually familiar with DNA. You've probably seen pictures in the media of this sort of spiral-shaped double helix kind of DNA structure. So we say DNA has a double helix structure. It's like a twisted ladder. So the double helix means it's kind of 2 strands that coil or wrap around each other. So DNA structure. DNA, or deoxy, see here I've got the older version, deoxyribose nucleic acid, is made up of 3 simple chemical units that are repeated millions of times. And that's called a nucleotide. So DNA is basically made out of nucleotides. Now, a nucleotide itself is actually made up out of 3 chemical groups. It has a phosphate group, a sugar group, and a nitrogenous base, some often just referred to as a base. So DNA is made out of this unit of these 3 chemical groups repeated millions of times. So a nucleotide consists of a phosphate, a sugar, and a base. So the phosphates and the sugars here are linked end to end. So the phosphate here will connect to another sugar, and the sugar down here will connect to another phosphate group. And so you get the phosphates and sugars are chained kind of end to end and form the backbone of the DNA, whereas the bases form kind of the steps of the ladder, the bits that actually hold the 2 strands together. So DNA is double-stranded, where you've got a sugar and phosphates running down each side, and the bases sticking into the middle. So it looks kind of like a ladder, but remember it's actually a twisted ladder, a double helix, each side of the ladder, so like a spiral staircase. So it's long sequences of this DNA stuff is what's actually coiled up into the chromosomes. You may have heard of chromosomes, are basically a DNA sausage. And these DNA sausages can be found in the nucleus of any animal and plant cells. So every living thing has DNA. It's not always organized into chromosomes quite this way, but in more advanced organisms, things that aren't bacteria, so like animals and plants, even fungi, you've got DNA organized, coiled up into these chromosomes, into these sausages that can be found in the nucleus of each cell. So let's look at how does this relate back to that structure of a nucleotide that we just saw. So that's kind of what we're trying to show here. So we said DNA is made up of the bases, and the two strands are actually twisted around each other, giving it that double helix. So this is the double helix version. So you can imagine if we untwist that, it would look kind of like this. So you can see this grey strip here is the grey spiral on one side, and this grey strip is the opposite, the grey spiral on the other side. So you can imagine if we sort of flattened this out or uncoiled it, it would look like this. So within the sides of the staircase, the grey stripes, you've got the phosphates and the sugar, so we call that the sugar-phosphate backbone, and in the middle we have the bases. So those make up the steps of the ladder. So the bases form base pairs. Now, if we look at the structure, so we've got a phosphate there, a sugar, and the bases form the steps. So a nucleotide, as shown kind of here, is one unit of those. So when we talk about, most textbooks are in a test, if they ask what DNA is made out of, the answer they're often looking for is nucleotides. So try to think of it like this. DNA is made out of nucleotides, pieces like this, and nucleotides are made up of a sugar, a phosphate, and a nitrogenous base. So think of it kind of in that order. DNA is made up out of nucleotides, these units, and a nucleotide is made up out of a sugar, a phosphate, and a base. We have, those of you who do a bit of chemistry, the lines here all represent covalent bonds. So there are very strong covalent bonds holding the backbone together. However, in between the two strands, we have weak hydrogen bonds here. And adenine and thymine, A and T, form two hydrogen bonds. You can see here, whereas guanine and cytosine actually form three hydrogen bonds across that space. And that means that the guanine cytosine, the GC bonds, here are slightly stronger than the AT regions. So in regions where DNA has to be opened, pulled apart, and we'll talk about that later, why that has to occur. Often it's easy to at least start that process in a region that's rich in A's and T's, because if there's lots of A's and T's to pull the strands apart, to break these bonds, it's going to be a little bit easier than if there are lots of G's and C's, because here you've got three hydrogen bonds versus the two hydrogen bonds between the A's and T's. So, the base pairing rule, sorry, before we get into that, there are four different types of bases, which we've already mentioned. So, adenine, which is often just referred to as A, thymine, which is T, cytosine, C, and guanine, G. And so, the base pairing rule is basically, and we'll talk about why this actually is in a sec, that adenine always pairs with thymine. And the easiest way to remember this is if you look at A is a letter that is made up of straight lines, pairs with T. T is a letter that is made up of straight lines, versus C and G, cytosine and guanine, are actually curved line sort of letters. So the curvy ones pair together and the straight line ones pair together. Now, obviously, sort of and vice versa. So A pairs with T, T always pairs with A. It doesn't matter which way around, but they always pair together. So one common question that they might ask in an exam is, for instance, they might not give you the letters on one side. They might not tell you what the letters are on say, this side. And they might give you the bases on this side and ask you to say, what would the sequence be here? Or they may put that in words. They may say, a double-stranded piece of DNA has the sequence, here, A, G, T, A, C, G on one side. What would the complementary sequence be on the other one? So you simply go down and you say, well, if we've got an A here, this must be a T, a C, an A, a T, a G. Always just the complementary, the opposite one that it would pair with. Okay, let's move on. So here's another representation. Maybe that makes it a little bit clearer. You can see here that lovely double helix, and here flattened out at the end to show you again those parts. So the parts you'd be expected to recognize on a DNA structure would be a phosphate, a sugar. So you've got the phosphate here, for instance a sugar here, a nitrogenous base. You could say a base pair is the pairs in the middle here, and a nucleotide. So we've got a phosphate, a sugar, a base, and a nucleotide, as well as possibly, I suppose, the sugar phosphate backbone. That's probably a little bit. It's not very common that they'd ask for that, but the sugar phosphate backbone's pretty simple too, just the bit down the side. Okay, as well as the term used for the structure of DNA is a double helix, so worth noting that one too. So, if we look at the nucleotides, if we look at their chemistry just a little bit closer, I'll just zoom in here a little bit. If we look at the structure of the nucleotides a little bit closer, they have two slightly different structures. Now you don't need to know the structural formula for these, but they are often drawn as these shapes. So the pentose sugar that DNA is made out of is often drawn as a pentagon. Now it has an oxygen here. It's not actually a carbon at the top, but the sixth carbon sticks up here. So the carbons are actually always at where the intersections. So there's one carbon here, 1, 2, 3, 4, and the fifth one sticks up here. So 5 carbons means it's a pentose sugar. And the phosphate's often just drawn as a circle. It is actually, phosphate is actually a phosphorus atom with oxygen atoms attached, but often just simplified to a circle. And then the base is also attaches on here, is also has this hexagon shape to it, and it is a ring that at these intersections you've got carbons, and possibly at one point being a nitrogenous base, a nitrogen atom too, I think. And again, you don't need to know the chemical structure. You can look it up, and you'll find it on Wikipedia. But the important thing to note is that some of these have this single ring structure. So you've got a single ring here, whereas some of the bases have a double ring structure. And you might be wondering, well, why is this important, or what has this got to do with anything? Well, it actually is important to ensure that correct base pairing. So we call these single ring structure ones, we call these pyrimidines. The best way to remember this is it's back to front. So the pyrimidine is the long name, and it's the single ring, whereas the purine is, they had to get it back to front. The short name is the longer one, the double ring base. So pyrimidines and purines. So cytosine, C, and thymine, T, are pyrimidines, whereas guanine and adenine are purines. And so you may sort of think, I would have thought, A and T, because they pair together, would both be... Well, think about that for a second. So we've always got one member of each pair as a single ring, has a single ring base, and the other member of each pair is a double ring base. So this is one thing that helps to ensure correct base pairing. Because if you had a cytosine and a thymine pairing together, you would have two single rings spanning that space in between the DNA. It would kind of be too short. So the sides of the steps of the ladder, if we had sort of one side of the DNA here. If we have a single ring base sticking out here and a single ring base, so a pyrimidine sticking out on both sides, well, you kind of end up with a gap in the middle. They don't completely span the gap. Whereas a purine, if you had two purines, well, kind of your two bases would kind of stick out too far, and they would kind of either overlap or actually force the DNA to bulge outwards, because it would be, the step there would be too long. So this is one thing that essentially the geometry of this helps to ensure that you always get a pyrimidine, a C or a T, pairing with its respective partner, the purine, a G or an A. So this helps to ensure that C pairs with G, and it helps to ensure that T pairs with A. So that's something worth noting. Now, it's unlikely you'd get a question on this, but if you're going for top marks, it may be something you want to add to your repertoire. So if we look at one other key point here, is we say that DNA is antiparallel. Now, what does antiparallel mean? Well, antiparallel means two lines that I remember hearing a really historic definition of parallel, which said, two lines that never intersect, even all the way to the feet of God, or something like that, which I thought was kind of funny. But essentially, parallel are two lines that never deviate, so they never get further apart and they never get closer together. So like train tracks are parallel. The two sides of each track never cross over and they never get further apart, otherwise the train would come off. So the sides of the DNA are parallel. Now they're twisted, but they stay the same distance from each other, so they are parallel to each other. But we actually say they are anti-parallel. So they are parallel, but running in opposite directions. So these two lines here are anti-parallel. They'll never cross over or get further apart, but this one is running in this direction and this one is running in this direction. So why is it that it's anti-parallel? Well, if you look at the structure of DNA here, and I might actually just pop back to one of the diagrams we had earlier. If you look at the structure of DNA here, you'll notice that on one side, and if you've made a little model up in class or something, you'll notice on one side that on this side, for instance, all the sugars, now the sugars always, to me, look a little bit like a house. That pentagon shape looks like a house. So the houses are always pointing upwards. The roofs are on top. Whereas if you look on the other side, the sugars are all upside down. So the sugar, that pentagon-shaped sugar, has a roof that faces kind of downward. So they're all pointing down this way. And that's why we say that the DNA is anti-parallel. So it looks like on this side, like all the little houses, the sugars are pointing up, and on this side they're all upside down. They're all pointing downwards. So that's why it's anti-parallel. So if in class you may have done a little activity where you piece together a sequence of DNA using these nucleotides, and you'll have noticed that on this side you have to actually take your little nucleotide like this, and you actually have to flip it upside down, and then so the nucleotide on this side is actually the same as this one here, just flipped upside down on the other side. So they're all upside down on the other side. Okay, so let's get back to where we were. Okay, so this is also important, because when we talk about DNA, we often talk about DNA having direction, or we have a way of labeling the direction. So you'll notice here we've said on this side of the DNA, this is the 5' end. And 5', they often just do a little 5 with a little dash to denote 5'. And this is the 3' end. And on the other side of the DNA, it's the other way around. You've got the 3' up here, and you've got the 5' over here. All right, so you've got the 5' here and the 3' here. So that's probably all you need to know. But I do feel sort of, as a teacher, I do feel it's kind of necessary to quickly explain why they chose those 5 and 3'. although this is probably going beyond what you would need to know, or you wouldn't be assessed on this. But it's actually very simple. If you look at the pentagon, when it's sitting there, the roof of the house here is the oxygen. So that oxygen, let's call that 0. O, round, 0, fair enough. And then the first carbon, if we go around clockwise, sits here. So we say that's carbon number 1. And then here, we've got carbon number 2. And then here, at this point here, carbon number 3, carbon number 4, and as I mentioned, the fifth one sticks up on a little stick up there. So we basically just number the carbons as we go around. So on the other side of the DNA, you've got the oxygen of the roof is pointing downwards, so the oxygen's there, and we can still go around clockwise. We've got carbon number 1, carbon number 2, carbon number 3, carbon number 4, and carbon number 5 sticking down this time. So that's how they've actually numbered it, because it means that what happens is at one end of the DNA here, so you've got again carbon 0, carbon 1, carbon 2, and then carbon 3. So sticking down from carbon 3 is always an OH group, or a hydroxyl group. And this is where another nucleotide can be added on. So nucleotides are always added if we were making this bit of DNA longer, or copying a bit of DNA. Nucleotides are always added to the 3' hydroxyl end, so this OH group on the third carbon of the end of the DNA, or the the strand of DNA. So the 3' hydroxyl is where, or the 3' OH group is where the next nucleotide always gets added on. And we'll look more at that. We'll mention that again quickly when we look at how DNA is copied, or and those sorts of things. So at the 5' end, we always have the phosphate group sticking out. So we can talk about the 5' phosphate and the 3' hydroxyl end. And so on this end, you can see here the OH, everything's just upside down. So the 3' OH group, or hydroxyl group, and down here the 5' phosphate group. So all you really need to know is that DNA is antiparallel, runs in opposite directions, and we've got a 3' and a 5' end. And on the other side will be the other way around, 3', 5' end. Cool. So, just before we leave, I thought it's also worth mentioning, and again, this is probably a little bit extra. I guess there are a couple of details in here you should know. But DNA is essentially compacted or coiled up into these chromosomes. And I thought I'd just explain a little bit more about how exactly that happens. Basically, you start with a strand of a double-stranded bit of DNA, and that wraps around proteins called histones. So DNA isn't normally found naked as just pure DNA. We call that naked DNA. In a cell, especially a cell that has a nucleus, DNA is associated with proteins called histones. So we find in the nucleus is actually full of a material called chromatin. So chromatin is actually just a combination of DNA and histones. So chromatin is basically what chromosomes are made out of. A chromosome is actually made up out of DNA wrapped around these little histones. So if you've ever done sewing, those little bobbins, those little metal kind of rings that threads are wrapped around, histones look a little bit like those. So you've got a strand of DNA here, and then that strand is wrapped around these histones. And you get this sort of like what looks like almost like a pearl, like a chain of pearls, as the DNA is wrapped kind of around the histones. And then those are coiled, and maybe if you imagine if you looked at that side on, it would sort of spiral out towards you like that. Those are coiled up into a strand of chromatin. So chromatin is just made up out of DNA and histones. So that forms a long folder, a fiber. So this thread here, you can imagine, forms a long fiber, because that is still an incredibly long molecule. And that fiber is folded in loops like this. And those loops are actually looped around in a flower pattern. So if you looked inside one of these loops, it would actually be a spiral of DNA wrapped around those histones. And those loops are kind of stacked one on top of each other, and that gives you this bottle brush like looking structure like you have here. And if you keep stacking them on top of each other, eventually you end up with what looks like a sausage. And that's basically your chromosome. And chromosomes are usually only visible under the microscope when they're being copied. So you essentially have a double sausage. You have two copies. So each copy there's one copy that I've circled. And each of those sausages is just a bunch of DNA coiled up around those histones. It's made up of those chromatid, out of that chromatin. So we'll come back and look at chromosomes and how they're important during DNA replication when DNA is copied before cell division. But we'll do that in one of the other tutorials. So I just thought I'd mention that here, that DNA is coiled up around that. So I think we're almost at the end. So here, just mentioning that so before replication you get the single sausage, and then usually what you see under the microscope is a double sausage chromosome. And so this is actually called a chromosome, and then this is called a chromosome. And one half, which is actually the same as what we started with over here, is actually called a chromatid. So chromosome is a bundle of DNA. Here you've still got a bundle because it's all joined together, but each half is a chromatid. And we'll mention that again when we're looking at more detail at chromosomes. So lastly here, it's starting to look at what does DNA actually do. So you may think, well, just a bit of chemistry and all boring structural stuff. But this is probably one of the most significant discoveries of the last century. And the reason I say that is it may not sort of seem so fundamentally important, but it is such an amazing discovery, because right back even from when Chargaff worked out those ratios, they were suspecting that DNA may be what codes for essentially our characteristics. So our DNA is what makes us who we are. And so the sequence of bases on the DNA, those A's, C's, T's, and G's, are what actually code for the proteins. And if you could go watch my video on what proteins are, but proteins are the moving working parts of a cell. So the enzymes, the receptors, and they're really what make the cell function. So if you think of a cell as kind of like as a building, the organelles might be the rooms within the building. Proteins are basically the workers. So the moving working parts in the surface of the organelles and those membranes, and inside each organelle, those things are proteins. And so essentially our DNA is the recipe for our proteins, and our proteins are the gears and cogs, the workers, the moving working parts of the cell, and they give our cell its function, and it's our cellular function that makes us who we are. So essentially, if you look at the person next to you, or look at anyone, they are really just the complex interaction of thousands of cells, and inside or millions of cells, and inside each of those cells, that cell is really just the complex interaction of thousands of proteins. And those proteins are all expressed or made from DNA. DNA is basically the recipe for those proteins. So the cell obviously needs some machinery to read these recipes and actually make the proteins and things. And we'll look a little bit more at how that works in some of the next tutorials. So just before we go, only parts of the DNA actually have sequences, so the A's, T's, C's, and G's that code for protein, and those are called genes. And that's why you may have heard of so some bits are non-coding and some bits are genes, where there are sequences, A's, C's, T's, and G's, that code for those characteristics. And if you changed that sequence, you'd get a slightly different characteristic, or that function may be lost, which would be a mutation. So it's that sequence of bases. But what was so incredible about this discovery is that not only do we have essentially the code of life, the recipes for, if I look at your DNA, if I take a copy of that, I essentially have the recipes to make another copy of you, to make a clone. So not only had we discovered the code of life, but the thing that was sort of more fundamentally kind of mind-blowing was that every living thing, in every living thing, this code of life was written in the same language. So the DNA in every living thing has A's, C's, T's, and G's: adenine, guanine, cytosine, and thymine. You don't get any other. You don't get other letters. You don't get. So it's simply the sequence. It's just the order in which that those are written. The sequence that makes us different. So it's like a computer. If you've ever done any computer coding or computer, those languages. There's lots of different languages like Java and PHP and C++ and geez, I don't even know all of them, but HTML. There's all these different codes or languages within which you can write a computer program or a web page. But in nature, in every living thing, the code of life is written in the same language. And that is kind of, the potential for what we can do with that is almost limitless. So for instance, there's automatically straight away, scientists had this idea, well, this technically means that we can take a gene from one living thing and put it into another living thing. And if it's written in the same language, that the other living organism should be able to express that. And indeed it does. So, scientists can have been working on, it's genetic engineering, taking the gene out of one living thing and putting it into another, so that it then also has that function or that characteristic. And as technology progresses, we may not even have to use existing recipes. We're actually starting to develop completely artificial genes, so genes where the recipe really has been written, by us to produce a protein with a certain function. Now at this stage it's still in its early days, where these artificial genes are usually just combinations. They've taken parts of the recipe for one protein and parts of a gene, the recipe for another protein, and kind of put those together. But in the future it may be possible to really write from scratch a recipe for a protein that carries out a function, some desirable function. As well as some people suffer from diseases because they have genes where there's some fault in that sequence. That sequence isn't so desirable. So there's also the potential that we may be able to correct those diseases or disorders by correcting the sequence to the best functioning version that's available. And so there's lots of potential, and this is definitely going to change the world around us in the near future. And it's a very exciting time to be in biology because of this, because there's a lot of discoveries going on. I often look at chemistry and think, oh geez, all the interesting stuff's already been discovered. I mean, I'm sure that's not true, but the really big fundamental stuff, it seems, has already been done. Whereas in biology, we're still working out. We've got the fundamental stuff there now, this DNA and how it works, but there's still a lot to be learnt there and discovered. And especially with the application, how we can actually now use this to treat disease, how we can use this to grow crops that are resistant to pesticides and or pests. All those sorts of things is still being worked out. So it's an exciting time to be in it. So the next couple of little video clips just show you, just briefly go over the structure again, which it's actually a little video presented by Watson, one of the two people who are credited with the discovery of the structure of DNA, and a little summary there on the structure, I think, as well. So worth watching. But keep that in mind, that it is really so amazing that the code of life in every living thing is written in the same language. And that is also a piece of evidence that suggests we did all evolve from a common ancestor. Okay, well, we'll leave it there and we'll get on with the next one.